So when we start to use these different drugs, there's a number of clinical considerations that we have to take into account before we do administer them. So ideal quantities for antibiotics, things that they would have is, first of all, we need them to be readily available. So this needs to be something that we can produce fairly easily. It needs to be able to be stored fairly easily. Okay, so readily available. It should also be pretty inexpensive. Chemically stable. We don't want something that is only going to last for a couple of days. We need it to last for a period of time so that we'll have time to manufacture it, ship it to the different doctors' offices and hospitals that are going to use it, and then they need to be able to keep some on hand there. So it does need to have a pretty good shelf life. It needs to be fairly easy to administer to a patient, and it should be non-toxic. It should be non-allergenic. We don't want to negatively impact the host that we're actually giving this to, because again, we're trying to help them to get better, and so we don't want something that's toxic and allergenic. And then a last thing that we want is that we want it to have selective toxicity against more than one thing. So we want its selective toxicity to work against a wide range of pathogens. So the spectrum of action gives us an idea of just how many different pathogens a specific drug is effective against. And this diagram right here just shows us a number of common drugs and exactly which types of microbes they can actually work on. Some of them only work on a few, just a select few, and then we have others where you can see the bar is much wider. It does cover a larger range of organisms, and this is what we call narrow spectrum drugs versus broad spectrum drugs. The narrow ones, they can only work on a select few. When we're talking about broad spectrum, these have a wider range of organisms, and so they're a little bit more useful because we can just kind of keep them on hand and use them for a number of different microbes. If we talk about effectiveness, we need our drug to be effective, and there's a number of different tests that can be used to determine exactly how effective a drug happens to be. So one of the tests that's commonly used is called a diffusion susceptibility test. This is also called a Kirby-Bauer test. And what this one does is it takes a paper, which essentially has the drug on it, it places it on a petri dish, and you can see a number of those here in this image, and then we look at the ability of the bacteria to grow around that specific drug. And what you can see is that in some cases there is lots of bacterial growth around the paper, and then other times we have a tiny little ring where they were not able to grow. Sometimes we have a larger ring where they are not able to grow. That ring of no growth is what we call the zone of inhibition. So what that tells us is that that is actually where the drug has diffused to. So this gives us an idea of how far the drug is diffusing, and that's affected by molecule size, molecule shape. Those are things that do impact the diffusion. We can rank different microbes as being susceptible, intermediate, or resistant to a drug based on this test. So if we have microbes that basically there's no ring, this would be something that looks resistant because there's no effect. We can have some that are kind of intermediate, which would be this one where you have some growth, it's not as far away. And then we have some that are much more susceptible, which are going to be the larger rings that we have right here. We can also use what's called the minimum inhibitory concentration test, or the MIC test. This is a measure of potency, basically how much drug do we have to use. We get the smallest amount of drug that will inhibit growth and reproduction of the pathogen. So what we do is we prepare a number of tubes with different concentrations of the drug in it, and you can see down here on the bottom that this image is showing us increasing concentrations of the drugs. We inoculate the little test tube with bacteria, and we see if we get any bacterial growth. And if it's cloudy or turbid, we can call it, we know that we did have bacteria grow there. So those are not high enough concentrations. And then if you just look starting right here and over, all of these show no growth inside the tube. So this is going to show us what concentration, what the minimum concentration is where the bacteria is not going to be growing and reproducing, but it doesn't necessarily tell us that there is dead bacteria there in that tube. So there may still very well be some living bacteria inside the tube. They're just not able to replicate. So we can use this test along with what we call an E-test. And the E-test shows us kind of the same thing, but it shows us all in one place. We've got a paper you can see in the Petri dish here. It has a range of drug concentrations on it, and we're trying to see at exactly what drug concentration do we stop getting growth around our paper. And you can see in this one, it's about right here. Whatever the concentration of the drug is right there, we're seeing no growth, okay? Because again, that's where we start to have this empty ring here where we have the clear Petri dish. The minimum bactericidal concentration test is going to tell us basically how much do we need if we want to kill the pathogen. So not just inhibit the growth, but kill it. And that is what that cidal part means on the name here, would be that this is something that actually eliminates the pathogen. So in this case, we run the MIC test, the MIC test that we just looked at. We take those clear tubes that we got, and we actually take what's in them and we plate it onto a Petri dish. And then we incubate them and we see, do we have bacteria that grow or not? And so you can see that it is possible to get a concentration where you don't have growth inside the tube, but if you go and you later plate those cells out, they're able to grow again. So that's not something that's bactericidal. The 16 micrograms per milliliter here, this is going to completely eliminate future colony growth. So that would tell us that that is the minimum bactericidal concentration. So if we want to kill the bacteria, we need to use the 16 micrograms in this example. So it is important to understand the difference between bacteriostatic versus bactericidal. The 8 would be bacteriostatic because it did inhibit the growth, but it didn't kill them because they grew later. And the 16 is bactericidal because it kills them, but then it also, they're not going to grow later because they are dead. We talk about routes of administration, how we're going to administer the particular drug. There are different ways that it can be administered. We can have direct application. Direct application could be called topical or local administration. So this might be some type of a cream that we're rubbing on the surface, so on our skin, and you've got some athlete's foot cream here, so that's the way that that would be applied. We could also have situations where we take pills, okay, so oral administration. And with those, they are the simplest mode of administering drugs, and some of the downsides of that is that there's going to be lower drug concentrations in the body. And another one is that there's very possible lack of compliance. So just think about how often people do miss their doses, or they forget to apply the medicine. Maybe they don't space them correctly, or they don't take them with food as they should. So when we're taking oral medications, or we do have the topical application, you do run this risk that the rules of how it should be administered are not going to be followed perfectly. Other ways that we can administer different drugs is it can be intramuscular, so IM, or intravenous. So these are either inserting it into the muscle, so that would be with a shot, or we could have an IV that's used. And you can see here what happens with the drug concentrations in the blood depending on the way that the drug is administered. You definitely don't get as high a concentrations if you look at the top diagram if we're using an oral drug. If we do intravenous, so IV, then we get a very high increase in the concentration in the blood. So you can see that it goes up and it stays nice and stable while we're giving the IV. And then if we're looking at the intramuscular, it does get delivered pretty quickly, but then it does taper off after a little while. We must also consider at the same time that the chemicals that we are putting in there for treatment, they might also be removed or inactivated by the body. So we have to keep that in mind. We have to think about how fast they're going to last just with our own body systems removing the typical toxins. Toxicity is important when we're talking about drugs. They may be toxic to the microbes, but they might also have toxic impacts on us, so negative impacts on us. And sometimes they are things that we might think are not too terrible. Here we have an example of some teeth discoloration. The enamel on the teeth is discolored. So the individual may survive just fine, but there are some negative aspects there. So safety of a drug can be calculated using the drug's therapeutic index, and we call that the TI. This is the ratio of the drug dose that can be tolerated to the drug effective dose. That's going to be the therapeutic index. If we talk about allergies, we want drugs that are going to be non-allergenic. And we also need drugs that are going to not disrupt the normal microbiota. We have a lot of microbes that have natural beneficial relationships in our bodies, and if we harm them, then that can create a disbalance. If we kill off some of them, that can allow for the overgrowth of others, ones that typically are kind of held in check. They get kind of extra nutrients and they're able to then cause diseases. If we talk about resistance to microbial drugs. If we talk now about resistance to antimicrobial drugs can be a real problem. And this resistance can certainly develop in populations, and this is something that we do see quite commonly. Bacterial resistance tends to develop in two main ways. Basically, we can have new mutations of chromosomal genes, and then the second way is that bacterial cells can acquire resistance genes or that information from other cells. So they can get DNA from horizontal gene transfer. So that would be by conjugation, transduction, transformation. All of those methods of horizontal gene transfer could allow a bacterial cell to acquire a gene that's going to make it resistant. So if we look at this diagram here on the bottom of this slide, we have a population of cells, and those cells already have some genetic variation there, and we can see that in the different colors. There happen to already be some cells that are resistant to the drug that we're going to use, and so when we do expose the cells to that drug, almost all of the cells die off because they are susceptible, but you do have the resistant ones that survive. And they survived, they will then multiply and reproduce. And then if we look at later generations, we're going to find that our later generations, they do have a much higher percentage of resistant bacterial cells. Here's another look at it. Antibiotic resistance occurs because we are specifically selecting the ones that are already resistant. We're not creating new resistant bacterial cells, but we're killing off the ones that aren't resistant so that the only ones surviving are resistant. So we've got the same general picture here. The blue ones are susceptible, the red ones are resistant, and you can see that when we use our antibiotic, basically we've killed off the blue ones. They're not there anymore, which means that the red ones, the tolerant ones, they are going to reproduce and survive. And if we talk about horizontal gene transfer, they can also pass that information to other cells, which can enhance the resistance of neighboring cells. The mechanisms of resistance fall into 7 main categories. So if we talk about those, one way that we can have resistance is that the cells may have an enzyme that is capable of destroying or deactivating the drug that we're applying. So you can see here that in this case, there is an enzyme. It takes penicillin and it breaks open that ring. So when we break open the ring, the penicillin doesn't work anymore. So that's one mechanism. Another mechanism is that the cell may be able to slow or prevent entry of the drug into the cell. So it's blocking the drug from even getting in. We can have situations where they alter the target of the drug, and when they alter the target of the drug is no longer to attach or bind to that particular target. Shape is really important, so anytime we alter the shape of something, that affects interactions between molecules. And in this case, the shape that's being altered by the bacteria is our actual drug target. We can have situations where these cells' metabolic processes are altered. And so basically the cells are not going to use that pathway anymore. They may be able to abandon or kind of bypass certain metabolic steps that the drug would typically affect. They can also pump the antimicrobial drug out of the cell before it has an opportunity to affect the cell. So they actually get it out. So they detoxify themselves essentially. And then another one is that they can retard, so slow down the diffusion of the drugs. And if they slow down the diffusion of the drugs, that's going to slow the cell's metabolic rate. And so that would be what we have in biofilms. So in biofilms, in general, they're slowing down the diffusion of the drug. A last one here is that the cells can synthesize a protein that forms a protective covering around gyrase. That was one of the enzymes that can be affected. And so they basically put a protective covering around it so that the gyrase itself is not going to be negatively impacted by the drugs that we use. If we talk about different types of resistance, it falls into two main categories. We have what's called multiple resistance and then also cross resistance. The multiple resistance is going to be when there is resistance to 3 or more antimicrobial agents. So basically there is a cell that has resistance to multiple drugs that we might try to use. If we're talking about cross resistance, this is referring to when resistance to one drug is going to confer resistance to some similar drugs. So we will find that the microbe might be resistant to a specific class of drugs that may have very similar shapes. Okay, if we talk about retarding resistance, so slowing down that process where the cells are going to be resistant to the drugs that we use. There are several approaches that we can use to try to slow down that overall process. Okay, there's 4 main methods. The first method is that we can maintain a high drug concentration for an extended period of time. So basically finish out a dosage. A lot of times when people do take antibiotics, they take them until they start to feel a little bit better, but they don't necessarily finish the whole bottle. And this step right here, this method is trying to basically counteract that. They say, no, don't stop when you start to feel a little bit better. We want to maintain that high drug concentration so that we do truly kill off the pathogenic cells. Another way is that we can use a combination of antimicrobial drugs. So we try to use kind of a cocktail. So we use a mixture. Here we can see that in some cases you have certain drugs that are synergistic and we have certain drugs that are antagonistic. So synergistic means that they enhance the activity of each other, and then we have other situations where they actually inhibit the activity of each other. So synergistic drugs would be good to use. We should also try to limit antimicrobial use to when it's really only necessary. And this is something that you do see a lot of information about these days. There's many health campaigns where this is something that's highly promoted. Here's just a look at some different advertisements, some campaigns that have been put out by different organizations to basically say that if you don't need antibiotics, don't use antibiotics. And sometimes antibiotics are taken for things that they're not even going to be active against. So viruses in general are not affected by antibiotics. So it doesn't make sense that if you get a virus, you take a bunch of antibiotics to overcome it. So basically just limiting unnecessary use of antibiotics and saving them for the situations when we really do need them. And then another one is that, of course, new drugs can be developed. That is not an easy process. That is something that the drug companies are certainly working on all of the time, but there is this constant race, this constant battle that as new drugs are discovered and developed, that very quickly after they're put into use, we do end up with populations of resistant pathogens. So development of new drugs is certainly something we want to do, but it doesn't want to be our only approach. We do want to use the first 3 methods kind of as initial approach to fighting resistance in bacteria, and then when we have to, we use a new drug.